Hello everyone, I am Dr. Rangana and welcome back to my channel. In the previous video, we were learning the chapter 14 of NCERT Biology Class 11 and the title of the chapter is Breathing and Exchange of Gases. In the previous video, we had learned up to the respiratory volumes. Today, we are going to learn about the different pulmonary capacities and the rest of the chapter. Let's start. By adding up a few respiratory volumes as described above, one can derive various pulmonary capacities which can be used in clinical diagnosis. The first one, inspiratory capacity or IC. Total volume of air a person can inspire after a normal expiration is the inspiratory capacity. This includes tidal volume and inspiratory reserve volume. So we can say IC equals to TV plus IRV. Now let's understand this. What it's saying the total volume of air a person can inspire after a normal expiration. So after normal expiration, what is our capacity? Firstly, the normal inspiratory volume, that is the tidal volume, and next after a normal expiration, if we inspire forcibly, then that is the inspiratory reserve volume. So adding this two, we get the inspiratory capacity. Next comes the expiratory capacity or EC. Total volume of air a person can expire after a normal inspiration. This includes tidal volume and expiratory reserve volume. So we can say EC equals to TV plus ERV. Now let's explain this. After normal inspiration, what we can expire, that is the tidal volume that we had taken in a normal inspiration, plus the expiratory reserve volume which we can expel through forcible expiration. So the expiratory capacity is tidal volume plus expiratory reserve volume, that is after forcible expiration. Now comes the functional residual capacity or FRC. It is the volume of air that will remain in the lungs after a normal expiration. This includes expiratory reserve volume plus residual volume. Now think about a normal expiration. After a normal expiration, we can do a forceful expiration. So in a forcible expiration, the amount of air that is expelled from the lungs is expiratory reserve volume, and after a forceful expiration, the volume of air that remains is residual volume. So adding these two, we get the functional residual capacity. Next, vital capacity or VC. The maximum volume of air a person can breathe in after a forced expiration. This includes expiratory reserve volume, tidal volume and inspiratory reserve volume. Or we may say it is the maximum volume of air a person can breathe out after a forced inspiration. So in case of a forced inspiration, the total amount of air that we are taking in is tidal volume plus inspiratory reserve volume. Then if we expire this air forcibly, then the total amount of air that we are expelling out of our lungs is tidal volume plus expiratory reserve volume. So the total capacity of our lungs becomes, no, it's not the total capacity, it's the capacity of total amount of external air that we are taking in or we are expiring. That is VC equals to expiratory reserve volume plus tidal volume plus inspiratory reserve volume. Next comes the total lung capacity, that is the total capacity of lung, the total volume of air that the lungs can hold at any time. It is the total volume of air accommodated in the lungs at the end of a forced inspiration. So this includes obviously the residual volume and the vital capacity, which is the amount of air which we can breathe in or out in a forceful respiration. So TLC equals to VC plus residual volume, and if we break down the vital capacity, we get expiratory reserve volume plus tidal volume plus inspiratory reserve volume. So TLC becomes residual volume plus expiratory reserve volume plus tidal volume plus inspiratory reserve volume. So this is all about different pulmonary volumes and capacities. Now moving on, we are going to study exchange of gases. Alveoli are the primary sites of exchange of gases. Exchange of gases also occur between blood and tissues. Oxygen and carbon dioxide are exchanged in these sites by simple diffusion, mainly based on pressure or concentration gradient. Solubility of the gases as well as the thickness of the membranes involved in diffusion are also some important factors that can affect the rate of diffusion. Pressure contributed by an individual gas in a mixture of gases is called partial pressure, and it is represented as PO2 for oxygen and PCO2 for carbon dioxide. Partial pressures of these two gases in the atmospheric air and the two sites of diffusion are given in the table 14.1 and in figure 14.3. The data given in the table clearly indicates a concentration gradient for oxygen from alveoli to blood and blood to tissues. Now let's study the table 14.1. It is all about the partial pressures in millimeter of mercury of oxygen and carbon dioxide at different parts involved in diffusion in comparison to those in atmosphere. So first study about oxygen. When the respiratory gas is considered oxygen, the atmospheric pressure is 159 millimeter mercury. In the alveoli, the partial pressure of oxygen is 104 millimeter mercury. In deoxygenated blood, it is 40 millimeter mercury. In oxygenated blood, it is 95 millimeter mercury, and in the tissues, it is 40 millimeter mercury again. In case of carbon dioxide, the partial pressure in atmospheric air is 0.3 millimeter mercury. In alveoli, it is 40 millimeter mercury. In deoxygenated blood, it is 45 millimeter mercury. In oxygenated blood, it is 40 millimeter mercury, and in the tissues again, it is 45 millimeter mercury. If you notice, you can see in the deoxygenated blood and in the tissues, the partial pressure of oxygen and partial pressure of carbon dioxide both are same. In case of oxygen, in deoxygenated blood and tissues, it is 40 millimeter mercury. In case of carbon dioxide, both in deoxygenated blood and tissues, it is 45 millimeter mercury. And if you compare both the partial pressures in different parts of air, you can see in case of atmospheric air, the partial pressure of oxygen is much higher than partial pressure of carbon dioxide. Also, in case of alveoli, the partial pressure of oxygen is much higher than that of carbon dioxide, because in the alveoli, the exchange of gases is ultimately taking place. In deoxygenated blood, as the name suggests, it is deoxygenated. Obviously, the partial pressure of oxygen is lesser than partial pressure of carbon dioxide. And in oxygenated blood, as it is oxidized, so the partial pressure of oxygen is much higher than partial pressure of carbon dioxide. Again, in the tissues, as carbon dioxide is generated as a output of different metabolic reactions, the partial pressure of oxygen is much lower than the partial pressure of carbon dioxide. We are going to study this figure letter. Similarly, a gradient is present for carbon dioxide in the opposite direction, that is from tissues to blood and blood to alveoli. As the solubility of carbon dioxide is 20 to 25 times higher than that of oxygen, the amount of carbon dioxide that can diffuse through the diffusion membrane per unit difference in partial pressure is much higher compared to that of oxygen. Now, we are going to study this diagram. Figure 14.3, it is the diagrammatic representation of exchange of gases at the alveolus and the body tissues with blood and transport of oxygen and carbon dioxide. So, what we can see, in the alveolar air, the partial pressure of oxygen is 104 mm of mercury and partial pressure of carbon dioxide is 40 mm mercury. And the exchange of gases is taking place with blood. Through the pulmonary vein, the oxygen rich air is going to the heart, and through the pulmonary artery, the deoxygenated blood is going again to the lung for diffusion. In the systemic arteries are carrying oxygenated blood from the heart to different organs. In the body tissues, the exchange of gases is again taking place. In this oxygenated blood, the partial pressure of oxygen is higher than partial pressure of carbon dioxide. And the partial pressure of oxygen is 95 mm mercury and partial pressure of carbon dioxide is 40 mm mercury. In the body tissues, firstly the oxygen is taken up, different reactions are going on there, then the output of carbon dioxide is diffused out into the blood again. So the blood becomes deoxygenated. In this deoxygenated blood, partial pressure of oxygen is 40 mm mercury and partial pressure of carbon dioxide is 45 mm of mercury. Through the systemic veins, this deoxygenated blood is going to the heart again. And from the heart, pulmonary artery carries this deoxygenated blood to the lungs, where by simple diffusion carbon dioxide is expelled out from the blood. I hope up to this it's clear. Now let's study about the diffusion membrane. In the alveoli, where the simple diffusion is taking place, this diffusion membrane configuration is very much important. So the diffusion membrane is made up of three major layers. As you can see in the figure 14.4, which is a diagram of a section of an alveolus with a pulmonary capillary. So this is the alveolar cavity, or we can say it's the alveolus, and it is the blood capillary. So what are the diffusion membrane composed of? It is composed of three major layers. As you can see in the picture, the first one is the thin squamous epithelium of alveoli, or simply the lining of alveoli. It is the squamous epithelium of alveolar wall, which is one cell thick. Next comes the basement substance. The basement substance is composed of two things. First one, the thin basement membrane supporting the squamous epithelium and the basement membrane surrounding the single layer endothelial cells of the capillaries. So after the basement substance comes the endothelium of blood capillary. So I think it's clear. There are three major layers in the diffusion membrane, which is composed of the thin squamous alveolar epithelium, which is one cell thick. Next, the basement substance, which is composed of the thin basement membrane supporting the squamous epithelium and the basement membrane surrounding the single layer endothelium cells of capillaries, and then the endothelium of alveolar capillaries. In between them, however, it's total thickness is much less than a millimeter. It's about 0.5 to 1 micrometer, the total thickness of this diffusion membrane. Therefore, all the factors in our body are favorable for diffusion of oxygen from alveoli to the tissues and that of carbon dioxide from tissues to the alveoli. So, what are the factors that we have learned already? Firstly, it is a partial pressure gradient, or we can say the pressure gradient or concentration gradient of different component airs, mostly oxygen and carbon dioxide. Secondly, the solubility of carbon dioxide, which is much higher. How much higher? 20 to 25 times higher than that of oxygen. And nextly, the diffusion membrane is very much thin. We all know that for diffusion to happen actively, the membrane barrier should be very much thin. Next, we are going to study about the transport of gases. Blood is the medium of transport for oxygen and carbon dioxide. As we have already learned, through blood, oxygen is carried to the tissues, to the whole body, and again through blood, the carbon dioxide as an output is carried to the lungs again. About 95% of oxygen is transported by RBCs or red blood corpuscles in the blood. The remaining 3% of oxygen is carried in a dissolved state through the plasma. Nearly 20 to 25% of carbon dioxide is transported by RBCs, whereas 70% of it is carried as bicarbonate. About 7% of carbon dioxide is carried in a dissolved state through plasma. We all know blood is mainly composed of two substances, that is the blood corpuscles and the plasma. So, in the blood corpuscles, in the red blood corpuscles, some amount of both the gases are carried, and in a dissolved state through plasma, some amount of both the gases are carried. As bicarbonate ion, we all know the formula of bicarbonate is HCO3-. So, in this, the carbon dioxide is also carried, which we will study later. Now, the transport of oxygen. Hemoglobin is a red colored iron containing pigment present in the RBCs. Oxygen can bind with hemoglobin in a reversible manner to form oxyhemoglobin. So, when oxygen binds with hemoglobin, it becomes oxyhemoglobin. It is reversible, that means oxyhemoglobin can break down into oxygen and hemoglobin. Each hemoglobin molecule can carry a maximum of 4 molecules of oxygen. Binding of oxygen with hemoglobin is primarily related to partial pressure of oxygen. Partial pressure of carbon dioxide, hydrogen ion concentration and temperature are the other factors which can interfere with this binding. A sigmoid curve is S-shaped curve. A sigmoid curve is obtained when percentage saturation of hemoglobin with oxygen is plotted against the partial pressure of oxygen. This curve is known as oxygen dissociation curve. In the figure 14.5, we can see the oxygen dissociation curve. On the x-axis, the partial pressure of oxygen in millimeter mercury is plotted, and on the y-axis, percentage saturation of hemoglobin with oxygen is plotted. We can see it initially it increases with the increase of partial pressure of oxygen. The percent saturation gradually increases, and on the top of the graph, we can see with increase in partial pressure of oxygen, the percent saturation becomes almost static. So, this is a sigmoid curve. It is highly useful in studying the effects of factors like partial pressure of carbon dioxide, hydrogen ion concentration, etc. on binding of oxygen with hemoglobin. In the alveoli, where there is high partial pressure of oxygen, low partial pressure of carbon dioxide, lesser hydrogen ion concentration, and lower temperature. The factors are all favorable for the formation of oxyhemoglobin. Whereas in the tissues where low partial pressure of oxygen, high partial pressure of carbon dioxide, high hydrogen ion concentration and higher temperature exist, the conditions are favorable for dissociation of oxygen from the oxyhemoglobin. This clearly indicates that oxygen gets bound to hemoglobin in the lung surface and gets dissociated at the tissues. So, these four factors are related differently with oxygen dissociation and association. The partial pressure of oxygen is directly related to association of oxygen with hemoglobin to form oxyhemoglobin. So, the higher the partial pressure of oxygen, the higher the formation of oxyhemoglobin. In case of partial pressure of carbon dioxide, the relation is indirect. The lower the partial pressure of carbon dioxide, the higher the formation of oxyhemoglobin. Similarly, the reverse is also true. The lower the partial pressure of oxygen, that means lower the formation of oxyhemoglobin, that means the higher dissociation rate. I hope up to this it's clear how the oxygen dissociation curve works. Now, every 100 ml of oxygenated blood can deliver around 5 ml of oxygen to the tissues under normal physiological conditions. You have to remember this. Coming to the transport of carbon dioxide is carried by hemoglobin as carbaminohemoglobin, about 20 to 25%, the percentage we have learned earlier. So, oxygen plus hemoglobin is oxyhemoglobin, carbon dioxide plus hemoglobin is carbaminohemoglobin. This binding is also related to the partial pressure of carbon dioxide. Partial pressure of oxygen is a major factor which could affect this binding. When partial pressure of carbon dioxide is high and partial pressure of oxygen is low, as we see in the tissues, more binding of carbon dioxide occurs. Whereas, when the partial pressure of carbon dioxide is low and partial pressure of oxygen is high, as in the alveoli, dissociation of carbon dioxide from carbaminohemoglobin takes place. That is, carbon dioxide which is bound to hemoglobin from the tissues is delivered at the alveoli. RBCs contain a very high concentration of the enzyme carbonic anhydrase, and minute quantities of the same enzyme carbonic anhydrase is present in the plasma too. This enzyme facilitates the following reaction in both directions. This reaction is very much important. Carbon dioxide and water in the action of carbonic anhydrase forms H2CO3, which again dissociates into bicarbonate ion and hydrogen ion, that is HCO3 minus plus H plus. We have studied earlier that majority of carbon dioxide is carried as bicarbonate ion in the blood. This reaction is both way. So, if we start from the tissue site where the partial pressure of carbon dioxide is higher, and that will lead to more formation of carbaminohemoglobin. So, in the tissues where the partial pressure of carbon dioxide is higher, carbon dioxide diffuses into blood, RBCs and plasma and forms HCO3 minus and H plus ions. Through a reversible reaction, when it reaches at the alveoli where partial pressure of carbon dioxide is low, the reaction proceeds in the opposite direction, leading to the formation of carbon dioxide and water. This carbon dioxide trapped as bicarbonate at the tissue level and transported to the alveoli out as carbon dioxide gas. every 100 ml of deoxygenated blood delivers approximately 4 ml of carbon dioxide to the alveoli. So, every ml of oxygenated blood carries how much oxygen? 5 ml of oxygen. And every 100 ml of deoxygenated blood carries how much carbon dioxide? 4 ml of carbon dioxide. I hope the transport of gases is clear to all of you now. If you have any doubt, please write in the comment section. I will try to explain in a better way. Now the last and most important part of regulation of respiration. You will learn about this in the higher classes more. Regulation of respiration. Human beings have a significant ability to maintain and moderate the respiratory rhythm to suit the demands of the body tissues. This is done by the neural system. We all can experience this in our day-to-day life. The respiration that is occurring throughout day and night, throughout our life, we cannot actually feel it all the time because it is regulated by the neural system. It is not something we are doing deliberately. Okay. So, in the neural system, there are specialized centers which regulate the respiration. A specialized center present in the medulla region of the brain called respiratory rhythm center is primarily responsible for this regulation. Another center present in the pons region of the brain called pneumotaxic center can moderate the functions of the respiratory rhythm center. So, in the medulla region there is respiratory rhythm center which is maintaining the normal respiratory rhythm. Again, in the pons region there is pneumotaxic center which is moderating the functions of the respiratory rhythm center. So, imagine it like a chain of reactions. Neural signal from this pneumotaxic center can reduce the duration of inspiration and thereby alter the respiratory rate. Also, a chemosensitive area is situated adjacent to the rhythm center which is located in the medulla region, which is highly sensitive to carbon dioxide and hydrogen ions. So, increase in these substances. What are the substances? Carbon dioxide and hydrogen ion. Increase in these substances can activate this center. Which center? A chemosensitive center which is located adjacent to the rhythm center located in the medulla region. So, activation of this center in turn can signal the rhythm center to make necessary adjustments in the respiratory process by which these substances can be eliminated. Receptors associated with aortic arch and carotid artery, which carries the blood, also can recognize changes in carbon dioxide and hydrogen ion concentration and send necessary signals to the rhythm center for remedial actions. The role of oxygen in the regulation of respiratory system is quite insignificant. So, what are the substances that are most important in the regulation? That is the concentration of carbon dioxide and concentration of hydrogen ion. The chemosensitive area is located near the respiratory rhythm center which is located in the pons. What does the respiratory rhythm center do? It regulates the respiratory rhythm. So, the change in concentration of carbon dioxide and hydrogen ion concentration can activate the respiratory rhythm center which can alter the necessary changes. Again, there is pneumotaxic center which is located in the pons region of the brain. It can also affect the respiratory rhythm center which can reduce the duration of inspiration and thereby it can alter the respiratory rate. I hope this chain of events is clear to you. Now coming to the disorders of respiratory system. Firstly, asthma. We all have heard about asthma. Every one has someone known who is having asthma attacks. So asthma is a difficulty of breathing causing wheezing due to inflammation of bronchi and bronchioles. You have to remember The inflammation of bronchi and bronchioles causes asthma, and the symptom that we can see is wheezing. Next, emphysema. What is emphysema? Emphysema is a chronic disorder. That means it is developed over a certain duration of time. It is not a sudden attack. It is a chronic disorder in which alveolar walls are damaged due to respiratory surface is decreased. Why the respiratory surface is decreased? Because there may be some irritants coming through the inspired air, and that's why you can see one of the major causes is due to cigarette smoking. When you are smoking a cigarette, the different pollutants are actually inside your lungs, and it causes the alveolar wall damage. And due to the alveolar wall damage, the alveolar wall, as you have learned in the diffusion membrane, the alveolar wall consists the surface of the diffusion membrane. So when the wall is damaged, the surface area for diffusion is obviously decreased, so the respiratory surface is decreased and emphysema occurs. The last disease is occupational respiratory disorders. In certain industries, especially those involving grinding or stone breaking, so much dust is produced that the defense mechanism of the body cannot fully cope with the situation. Long exposure can give rise to inflammation leading to fibrosis. What is fibrosis? It is proliferation of fibrous tissues and thus causing serious lung damage. Workers in such industries should wear protective masks. As the name suggests, it is occupational hazard, so the safety measures should be taken. Coming to the summary of the chapter, breathing and exchange of gases. Cells utilize oxygen for metabolism and produce energy along with substances like carbon dioxide, which is harmful. Animals have evolved different mechanisms for the transport of oxygen to the cells and for the removal of carbon dioxide from there. We have a well-developed respiratory system comprising 2 lungs and associated air passages to perform this function. The first step in respiration is breathing, by which atmospheric air is taken in, that is inspiration, and the alveolar air is released out, that is expiration. Exchange of oxygen and carbon dioxide between deoxygenated blood and alveoli, transport of these gases throughout the body by blood, and exchange of oxygen and carbon dioxide between the oxygenated blood and tissues, and lastly utilization of oxygen by the cells, that is cellular respiration, are the other steps involved in respiration. Inspiration and expiration are carried out by creating pressure gradients between the atmosphere and the alveoli with the help of specialized muscles, intercostals and diaphragm. Volumes of air involved in these activities can be estimated with the help of spirometer and are of clinical significance. Exchange of oxygen and carbon dioxide at the alveoli and tissues occur by diffusion. Rate of diffusion is dependent on the partial pressure gradients of carbon dioxide, PO2 and rate of diffusion is dependent on the partial pressure gradients of oxygen, PO2 and carbon dioxide, PCO2, their solubility as well as the thickness of the diffusion surface. These factors in our body facilitate diffusion of oxygen from the alveoli to the deoxygenated blood as well as from the oxygenated blood to the tissues. The factors are favorable for the diffusion of carbon dioxide in the opposite direction, that is from tissues to alveoli. Oxygen is transported mainly as oxyhemoglobin in the alveoli where partial pressure of oxygen is higher. Oxygen gets bound to hemoglobin which is easily dissociated at the tissues where partial pressure of oxygen is low and partial pressure of carbon dioxide and hydrogen ion concentration are high. Nearly 70% of carbon dioxide is transported as bicarbonate ion HCO3- with the help of the enzyme carbonic anhydrase. 20 to 25% of carbon dioxide is carried by hemoglobin as carbaminohemoglobin. In the tissues where partial pressure of carbon dioxide is high, it gets bound to blood, whereas in the alveoli where partial pressure of carbon dioxide is low and partial pressure of oxygen is high, it gets removed from the blood. Respiratory rhythm is maintained by the respiratory center in the medulla region of brain, a pneumotaxic center in the pons region of the brain, and a chemosensitive area in the medulla can alter respiratory mechanism. Thank you for watching.